or the NF-kappa B here. And these mutants, the sequences, are added down on this line. And just of note, these plasmids have also been used in other studies for a number of years now. What we expected to see, if the IL-8 promoter actually depends on this transcription factor, when we compare the induction to wild type, we expect to see a decrease in the actual luciferase. If it doesn't depend on it, we expect not to see any change. Now with CBP here, we compare it to the wild type IL-8, and we don't see any drastic changes. Same with the IL-8 mutant NF-kappa B. When we compare to wild type, there's really no change. We were surprised to see with the mutant AP1, where we had previously seen that all the proteins, preME, NS1G, and NS5, inducing IL-8, also inducing AP1, we had expected some dependence and for these bars to drop. But instead, we saw an increase in the amount of IL-8 promoter-driven luciferase expression. And this was particularly significant with NS5 and preME. So going back to a little bit of the beginning of the talk, I showed that dengue virus, after 72 hours post-infection, activated the IL-8 promoter. And when transfected with any single protein, after 24 hours, we see activation of the IL-8 promoter. This alone, to me, suggested that the differential timing suggested that dengue virus has additional mechanisms regulating IL-8 activation. Now, of the three proteins that activate AP1, we saw that when we mutated AP1, instead of decreased, we saw increased expression from an AP1 mutant IL-8 promoter-driven luciferase expression. So we took a closer look at AP1. Classically, AP1 is an activating transcription factor, which can be homo or heterodimers of Jun family proteins, or heterodimers between Phos and Jun family proteins, or the ATF family and Jun family proteins. Within the Phos family, there are CPhos, Fra1, and Fra2, which under certain contexts have been shown to be inhibitory. So it's possible that we were actually producing both activating and inhibitory forms of AP1, leading instead of a strictly activating model to a modulated model of IL-8 activation by AP1. Therefore, we ask the question, which components of AP1 actually bind the IL-8 promoter during dengue viral infection? We used C-Jun here as a control because it had previously been shown that dengue virus induces C-Jun early in infection. Now, to measure which components of IL-8 bound or which components of AP1 bound the IL-8 promoter, with infected cells, we either mock-infected or infected cells with dengue virus, and then harvested at 0, 6, 24, 48, or 72 hours post-transfection or post-infection. We lysed these cells and loaded them onto columns with magnetic beams conjugated to the IL-8 promoter, washed away everything else, and came out with two fractions. The first being the protein that could bind the IL-8 promoter, and the second being the protein that could not bind the IL-8 promoter. Here I'm showing what bound the IL-8 promoter, either in dengue virus 2 infected cells or mock-infected cells. And ultimately, looking at C-Jun, Fra1, and Fra2, we didn't see any difference. There was no difference in the transcription factors, the components of AP1 bound to the IL-8 promoter. We used actin as a loading control from the flow through. So to summarize this portion of the talk, we asked three particular questions. First, what individual transcription factors are activated by dengue viral proteins? And We saw that pre-ME, NS1G, as well as NS5 also activated transcription from an AP1-mediated reporter. We saw trends towards dose-dependent activation with C and NS4B. Activation of the CDBP and NF-kappa B mediated reporters were both very low. So we used a looser statistical analysis that we interpret with caution, but conclude from that the CDP mediated reporter was induced by capsid premembrane envelope, the pre-ME construct, and NS5. And the NF-kappa B mediated reporter was induced by C, E, pre-ME, NS1G, and NS5. We also asked, does the activation of the ILA promoter by dengue viral proteins depend on any single transcription factor? And no, the ILA promoter activation by pre-ME, NS1G, NS4B, as well as NS5 did not depend on any single transcription factor. We were surprised to see increased activation from the AP1 mutant, and therefore we looked closer at the components of AP1 that could bind the ILA promoter, but we did not find any differential binding of AP1 subcomponents, c-Jun, FRA1, or FRA2, to the ILA promoter. The IL-8 pathway is highly regulated, as I mentioned before. NF-kappa B, the Jun N-terminal kinase, and P38 are three of the pathways most commonly associated with IL-8 activation. At the beginning of this study, we knew that NS5 induced IL-8 promoter activation and IL-8 secretion. This study has also shown that NS1G and NS4B also induce the IL-8 promoter. The previous study characterizing NS1G originally showed that NS1G only signaled in the presence of anti-NS1 antibody. And the discrepancy between this study and the previous study is probably because of the overexpression system that we use here. There is much less NS1G actually produced during the context of viral infection. In addition, we show that pre-ME also induces the ILA promoter, though it's not clear from this study whether that's from the actual production of protein processing of pre-ME within the cell, or whether the formation of viral-like particles and an external signal is contributing to the activation of the ILA promoter. We also show that the RONTES promoter is activated and TNF mRNA is produced by NS5. I've highlighted AP1 here today, as I've shown that many of the proteins that activate the ILA promoter also activate AP1. However, we weren't able to see any differential binding of AP1 subcomponents to the ILA promoter following dengue viral infection. So this remains an area to be studied. Other studies more recently have characterized other aspects of IL-8 activation by dengue virus and have shown that TLR3 is important. Mice cells do not contain TLR3, so this lends further support that there are multiple mechanisms activating IL-8. C has also been shown by one paper to induce IL-8, although I only saw trends towards dose-dependent activation. And what I suggest here is that perhaps it's the C-induced chromatin modification that is contributing to IL-8 promoter activation, opening up the promoter for binding by transcription factors induced by other viral proteins. In addition, mRNA stabilization does have to occur for IL-8 secretion Our previous study showed NS5 induced IL-8 secretion, so presumably this mRNA stabilization, which is predominantly characterized to happen through P38, does occur. And recently, as in the past 6 months, it was published that NS4B in a monocytic cell line will also induce IL-8 secretion. So it's possible that these two proteins are actually inducing P38 and mRNA stabilization of IL-8. In the wider context of pathogenesis, we have RANTES and IL-8 both recruiting T lymphocytes. TNF and IL-8 have been shown to act in concert to induce plasma leakage. one proviral role for it. But something that hasn't been characterized that I find rather interesting is that IL-8 actually will recruit, or can recruit dendritic cells, macrophages, and mast cells. All of them contain the CXCR1 or CXCR2 receptor. And therefore, this is a potential mechanism of recruitment of new target cells for infection. However, this has not yet been formally shown. So I'd like to close with my acknowledgments. Obviously, this slide does not have everybody who contributed to this work, but I would like to profusely thank Masanori for taking me on this past year and helping me bring this work to completion. Alan, who could not be here, definitely a brilliant advisor and one who has given me a lot more freedom than he probably should have.